Let me do a brief introduction of our speaker and conversationalist, or with whom I am in conversation tonight, Dr. Barbara Hahn. Barbara arrived at Cary Institute in the late spring, early summer of 2014. I know this because she arrived just before I did. I like to say we're in the same cohort. Barbara came after a couple postdocs at the University of Georgia's Odum School of Ecology, where she had funding from NIH, the National Institutes of Health, and NSF, the National Science Foundation. She did her PhD at Oregon State University, and if my memory serves me correctly, undergraduate degree at Pepperdine. Barbara is at the interface between ecology, computing, and public health. And that is a relatively new field. In fact, it's a very new field, and one which she has contributed to in many different ways. Barbara has worked with the WHO. She has worked with the NIH Centers for Research in Emerging Infectious Diseases, which she explained to me is a network of people on these issues, working on these issues. She's advised the Defense Advanced Research Programs. Oh gosh, and I forgot the A again, Barbara. You'll tell me when you come on. Agency.
Yeah.
There we go, Programs Agency. DARPA, among other things, are the people who brought us the internet. And she has looked at this working with many organizations. And before the last administration, she was an advisor to the Office of Science and Technology Policy, Pandemic Prediction and Forecasting Science and Technology Working Group. She's advised and been on councils at the National Academy of Science, and I like to say is extremely distinguished despite being relatively early in her career. So Barbara, thank you for joining us. And let's start off, if we could, with the first slide. And you know, this is one of those easy questions, but you know, disease ecology as a field isn't that old. Let's put it this way, it's young enough so I remember it being founded in the 80s. But can you tell me, how does a nice bat and frog ecologist who loves animals end up in disease ecology? What was your path to get you here?
Yeah, that's a good question. I remember my earliest memories is wanting to be a vet. And then as many of us with tiger parents might be able to connect with, I was quickly convinced to go into medicine instead. So that was pre-med actually when I started college. And I just really quickly got bitten by the research bug. Was really excited by the fact that we could ask and design studies to answer questions and discover knowledge that nobody else knew, to contribute to our global knowledge base. And then I decided to go to grad school, and I remember some of the earliest papers that I read that had to do with infectious disease in animals were written by colleagues of mine who are colleagues of mine now, Rick Osfeld and Felicia Keesing at Bard College nearby. They've written some of the seminal papers on infectious diseases and how animal populations and communities of different animals can influence infectious diseases in people, and the spillover process and the levels of those pathogens found in those communities. And I just remember I have like a cartoon image of in my head of like, stars going across, across my eyes. Or I just thought, I can't believe that this is an actual field. And of course, at that time, it really wasn't a field. It was a couple of really bright and creative scientists writing papers that were new, a new way of thinking about a system, and infectious disease being a major part of that system. And so I kind of never looked back after that. And that's how I ended up here. I just kept kind of pulling the thread of animals and what are animals doing? What is their ecology? Why does that matter? How does that matter for people? And somehow I ended up doing artificial intelligence research as well. So
we'll get to that.
A winding road.
Yeah, we'll get to that. But I think the vet route, I mean, I wanted to be a vet from the time I was 4 to 13 or 14. Then I realized, of course, that vets mostly deal with dogs and cats, and that led me into wildlife biology. But this idea of diseases as organisms and diseases as part of an ecosystem that then interact with the other pieces of that ecosystem, I think is a really powerful way of thinking about disease, which is not, if you become a doctor, you probably wouldn't have thought of it that way, right? So I'm glad you didn't stay, I'm glad you left pre-med and became a disease ecologist. So here's a question that we agreed was the softest of softballs when we were, talking about questions we might ask. But, so why is disease ecology so critical at this juncture in our history?
Oh gosh, yeah. I mean, where to start? Where are we right now? We're on Zoom. We'd ordinarily be doing this in person, right? And auditorium full of people, unmasked. Yeah, disease ecology, I think, is now surprisingly really front and center. It's been part of the national dialogue in a way that I have never seen before in my career. The reasons for that are obvious and, a little sad, but it's critical now more than ever because, it's always been critical. That's why we're in this field, right? We've been talking about infectious diseases and their impacts on systems for gosh, my whole career. And it's only been in the past 5 years that it's sort of started to ramp up. And of course, Ebola, Zika, and then SARS-CoV-2. It's just kind of been one after another. And it feels like it's not slowing down. We're in the middle of another Ebola outbreak right now in Uganda that we just got over monkeypox. Well, it's going down. Avian influenza is as big a threat as it's ever been. That was the leading worry for most of us about a pandemic prior to SARS-CoV-2, and it remains just as much a worry now as it was before, even after SARS-CoV-2. So disease ecology, yeah, that is a softball question. There's so many reasons to care about it. There's so many reasons why it's been proven to us that it's integral to the way we need to move forward as a society.
So Barbara, when you talk about avian influenza being a threat still, is that because of it potentially spilling over into people, or is it because of the impact it has on our production systems for livestock?
Yeah, good question. Both, I would say. Obviously, lots of poultry have been culled as a result of, flocks coming down with avian influenza. Lots of birds dying, but also the spillover risk to humans. I mean, the avian influenza is it's respiratory. I mean, it's going to spread fast if it gets into a human in a form that is highly virulent and spreads quickly. And so, it has all of the same characteristics as SARS-CoV-2, and it reassorts quite quickly. So
I'm just going to check. Leslie, I don't think we're seeing slides.
Josh, we're having some technical difficulties.
Okay, well, we'll talk about that then.
Yeah.
So that's good to know. We can do this without PowerPoint.
You
right?
certainly can.
Yeah, no big deal.
But Barbara, we talk about diseases that you study being zoonotic, right? Number one, what is a zoonotic disease? And number two, what proportion of the diseases that we encounter are indeed zoonotic, and if they're not zoonotic, what are they?
Yeah, great question. Zoonotic diseases, I feel like most of us have heard this term now, which is shocking because I usually have to really explain it and talk some things, but essentially zoonotic diseases are those that persist in animals and in many ways naturally reside in animals, oftentimes without causing any overt infection or disease symptoms, but can infect humans. They spill over to infect humans. The recent estimates in the literature say about 2/3 of diseases that are considered to be emerging in humans, so diseases that are growing in incidence or growing in geographic area, 2/3 of those diseases in humans are zoonotic, and the majority of them are zoonotic, and they range from, I think we can name a bunch of them just off the tops of our heads, right? We've already talked about Zika and Ebola, SARS-CoV-2, SARS, MERS, malaria. Yeah, I think malaria is now considered really human endemic, but there are malarias that are zoonotic, that are residing mostly in animal species. Yeah, there's just, we could just go down the list. And the frequency at which they kind of become part of the public dialogue feels faster, right? Like, I think all of us can attest to that.
Yeah.
In the studies that have looked at the increasing frequency of disease emergence events over time, even after you correct for things like the advent of the internet, where information about diseases spreads faster and becomes available much more easily, even after controlling for that, the incidence of these diseases and the emergence events has gone up since the 1980s. So I think this is a real phenomenon. Scientists agree that we're getting more of these zoonotic diseases emerging into human populations over time.
And it's, I mean, we can get to this, but I think that's in part because of the way we live our lives now.
Yeah,
Right.
there's
We're
a lot more.
interacting more, we're traveling more.
Absolutely. Yeah,
Yeah.
we're more connected than we've ever been. And I can't think of the statistic off the top of my head, but there's a way that we define remote, you know, like how long it takes to get to a place before that, you know, how far removed it is from civilization and from the rest of the world to how long it takes to get there. And I think that like a diminishingly small percentage of the world is considered to be remote now. So you can basically get anywhere on the planet within about a day or a day and a half.
Well, and we can communicate with anyone, anywhere on the planet pretty much in real time.
Yeah, it's pretty remarkable.
Except for if you're in parts of Dutchess County where we don't have cell coverage, but you know, that's exceptional.
That's right.
All right, so there, you know, the traditional way of going out and studying the distribution of diseases was to go out and sample and do some more samples and bring your samples back to the lab and run them in the lab, and you could do very sparse coverage without a lot of density of information, and then you had to extrapolate wildly from that. You know, we have new and different approaches, one of which is, as you say in the business, AI/ML, or artificial intelligence and machine learning. And I was wondering, you know, before we talk about the application of it, I think everybody's heard about machine learning and artificial intelligence, and they sort of have a vague idea of what it is. They know that, you know, some organizations use it to sell us things we might not need, but clearly want, and they know we want. But can you talk a little bit about what AI and machine learning actually is and how it intersects with our daily life?
Yeah. So the way that I like to think about machine learning and AI is analogous to how I think about human intelligence, human learning, and human intelligence, right? We're intelligent broadly, generally, because we know information from observing things and learning, training our brains to recognize patterns out of lots of data that we're taking in all the time. Those are kind of our neural nets. Our brains are a big, very dense neural net that's very complex. Machine learning and artificial intelligence share that same relationship, where machine learning underpins what we call artificial intelligence. Machine learning is learning those patterns from data. Oftentimes the machine learning is trained by a human. Sometimes it's asked to identify patterns that are suggested by the data themselves without the human having to identify what it is that the pattern of interest is. But in any case, the machine learning underpins that general artificial intelligence, which is this idea that a machine or an algorithm can make decisions and perform tasks without being focused or supervised by a human. So that is sort of the goal of AI research, or the holy grail. But there is a continuum, right? Sometimes when you ask something to a machine learning algorithm to predict something, that might be considered machine learning because it's in a particular use case.
Got it.
You can imagine that thing becoming a form of artificial intelligence if you're like not really telling it what it is you want it to do, but you're giving it lots of information. And it's a lot more information that you might need to give a human. But by giving it all of that information, you're kind of training its foundation. It's allowing the machine to do this foundational modeling that then can be extrapolated to situations that are outside of its normal domain or outside of the information that you gave it that was trained on. So there's lots of examples of technology that does this that we've all interacted with. Like you mentioned Amazon, like telling us that we should buy things that we don't want, but we do. And then there's music software, right? Like Spotify and Pandora that we listen to all the time, and Netflix, which recommends movies for you of interest. And gosh, what else is there? There's a really cool, I don't know if you guys have heard of Dolly. Do we have that on a slide, Leslie? There's an algorithm that you can say, like, you can give it a description of the kind of art that you want, and or something that you want an image of, and it'll just produce it. So you can just type into a little browser window, teddy bear and a decision tree and the style of Sendak, and it'll generate an image for you that looks like an artist could have, there it is. Yeah. Looks like an artist could have, actually created that, but that was an artificially intelligent, it was an AI machine that did that.
Right.
So
And
there's lots of examples.
there was an article recently in the Times about an art competition where the winning entry was an AI entry. And there was a wonderful discussion about how it is art, but it's a different kind of art, right?
Yeah, there's also the recipe creator. Have you heard of that
Yeah.
one? Yeah. I think there's a recent article about how to make your Thanksgiving meal entirely out of AI suggested recipes, which seems a bit risky, but.
Yeah, and I think, I can't imagine AI suggesting that you would put marshmallows and brown sugar on top of sweet potatoes.
That's crazy.
It just doesn't seem to be something that you could get a pattern recognition to tell you. All right, so let's go back to pattern recognitions. AI machine learning, can be harnessed to make predictions about many things. And one of the things you're looking at is whether we can harness it to make predictions about future pandemics. So how would you construct an algorithm, a formula, a set of pattern recognition software that will allow machine learning to be able to find that pattern and tell us what the next spillover event or next pandemic would be?
Yeah, initially this started out as just trying to answer a very basic question, which was, gosh, we have a lot of animals that carry diseases that can infect us. Can we look at all of the different kinds of data that we have available for each of those animals and try to train an algorithm to tell us the difference between those species that carry lots of diseases compared to those that don't seem to carry any. And so this is a sort of simplified diagram that you're seeing here on the slide that looks at a bunch of characteristics about each of these species and tries to train an algorithm to distinguish between the characteristics of something that's a disease carrier versus something that's not. So that's one really simple, that was the very first way that we try to apply machine learning to this question about infectious disease spillover and which species should we be concerned about? Are there ways that what can the algorithm suggest about these species that will allow us to then go out and test hypotheses in a smart way? And so the very first analysis that we did, here's some examples of traits that we train the algorithm on. We focused on rodents because they kind of have a bad rap of having lots of infectious diseases that can infect humans. And the algorithm suggested this pattern of species that have a fast life history strategy that kind of live fast, die young. They have large litters earlier in life. They have larger ranges. They, of course, overlap with humans. So there was a suite of features that kind of drew us a caricature of a species that is a zoonotic reservoir, a carrier of lots of diseases. And that, of course, led us down, suggested lots of hypotheses. Well, maybe it's something to do with their immune system, or maybe it's something to do with their patterns of spatial overlap with humans, or maybe it's something to do with the fact that when we cut down their forests and remove their habitats, they are still able to persist in and around human dwellings. So there's lots of suggested hypotheses that came out of this analysis that were confirmatory of things that scientists had looked at, particular systems in particular locations and found patterns. So it was confirmatory in a lot of ways, but it also opened up a huge new way of looking at these data across different species, across different large geographic range areas. We've done similar analyses now to try to understand what distinguishes bats that carry filoviruses like Ebola and Marburg from other bats that don't carry those kinds of viruses. We've done it for carnivores here to try to understand what distinguishes carnivores that are susceptible to these diseases. We've also done it for SARS-CoV-2. We did that early in 2021 and have been sort of trying to hang on to that runaway train for a while now, trying to keep track of the species and which variants they're carrying and which ones seem to be susceptible now that we didn't know about before.
Right. And so it's this sort of iteration of figuring out which species are susceptible and then looking at the different life history traits and then training the machine on those. And then
yeah, exactly.
So it's iterative, and the more information we get, the better we can refine our models. The more data we have about different life history categories, the better we can train our models.
Yeah, and these models are, they're powerful, but they're also limited. right? They're powerful in the sense that I see it as a way of extending your senses. We as field biologists are trained to go out and study a system and take notes and collect data with our senses or with the help of particular tools. They can measure things that we can't. And machine learning allows us to take those kinds of data at scale and also read in sensor data and also read in all kinds of data that you can't possibly go out and measure yourself and make sense of it in a multidimensional, high dimensional way.
Right. And so there are lots and lots of different variables. And I remember at one point you said to me, the real heroes of your work, right, are the generations of naturalists and field biologists and museum curators who have collected all these data over time. Can you talk about the process of how you collect those data, but also how you then put them into the model? Because it's really a two-step process.
Yeah, it's actually, and I stand by that statement. I think that, you know, the world is calling upon scientists more and more to make predictions that are actionable, that will help us to preempt issues that we're experiencing now with disease.
What do you mean?
The folks that the folks that contribute these data are field biologists and grad students who are, you know, going out and really just trying to uncover basic information about these species that we didn't know about before.
I have to load them
But then there's a bunch of species that
one by one.
Leslie, Lori, somebody's got to mute.
Oops.
There's somebody unmuted.
There's always got to be that one.
Yeah. All right. Go ahead.
Yeah, but I think I want to emphasize too that, you know, we talk about machine learning being a powerful way to sort of focus our surveillance efforts, but we still really depend on those surveillance efforts. Like the folks that go out in the field and catch the bats and test the bats. That is really incredibly hard work. And that stuff still remains underfunded despite the obvious need for those things. So I think machine learning, it really depends on the data, the quality of the data that you have. And you can't expect to get anywhere with machine learning and AI. And it's like it's a magic wand or something without the hard work of the folks that are collecting and curating and making this data available to the world.
Right. And I think that's critical because in a sense, you know, in the old days, we were looking for a needle in a haystack. And what AI machine learning do for us is give us a really powerful magnet, but we're still trying to find that needle, right?
Yeah.
And so it's a much stronger tool, but it still relies on, as you say, the people in the field doing hard work in some really remote places. And,
absolutely.
And there aren't that many of them, but that tends to be the place we send people off to collect these data.
Yeah, and it's really dangerous too. I mean, it's not like comfortable.
No.
Our way, it's expensive.
And so one of the things that your models do is help us identify, you know, you've got, 1,000 bat species, 800 bat species, and you know that Ebola is in bats, right? So we get that far, but your model helps us figure out which bats and why. And so how well are the models doing at learning this and coming back to us with better predictions?
Yeah, the models are doing all right. So this was the initial study that we trained an algorithm to recognize the characteristics of bats that had ever tested positive for a filovirus. Now, this was like scraping the bottom of the barrel in terms of data, because it wasn't like these species were confirmed to be carriers and replicators of these viruses. They were just seropositive, which means that they had an antibody response to virus.
So they've been exposed at the very least.
They've been exposed. And so it was really us trying to get a clearer picture that was really, really murky and not very data rich. So when we did it, the algorithm, it worked surprisingly well given the data that we had available to us. It was able to distinguish filovirus positive bats from negative bats with about 87% accuracy just based on their traits, which was remarkable. But I think the most remarkable thing is watching the predictions that the algorithm made become confirmed by independent groups. And so there was a group that was led by, one of the leaders of this team was Simon Anthony, who I believe is now at UC Davis. I believe he was at Columbia at the time, but he confirmed some of these species that you see on the slide as being positive for not only for filoviruses, but for a new filovirus. It's now called Bombali virus. And this one in China in 2017, that one was, I believe it was number 5 on our list of predicted species, which were rank ordered in order of their risk of carrying filoviruses. And, it was published 6 months after our study came out, and clearly they had been working on the system for months and months, if not years. And they sequenced this bat, which was, it's not in West Africa. It's not in Africa at all. Its range is in Southeast Asia, and it was found to carry a filovirus that was new, and it was found with a tropism in the lungs, which, it's not a great sign. You would rather find filoviruses in the GI tract or somewhere where it's not going to be like the aerosolizable, but it was found in the lungs. And so watching these predictions be confirmed is sort of a weird experience, right? Because you kind of want to be wrong, but you don't really want to be wrong, but you don't want to be right, but you're happy when you're right. I don't know. It's weird, but these have been confirmed.
I think that's been the issue with your predictions on SARS-CoV-2 as well.
Yeah.
Right?
Yeah.
Are we going to talk about that later, or should I talk about it now?
I mean, we can talk about it now. That was one of those analyses where we thought, well, we can't just sit back. This is like there's no data on SARS-CoV-2.
2.5 years ago, just after the pandemic.
Yes, right? It was we were locked down. My kids were freaking out about school not being in session, and I'm sitting in my home office, my new home office, trying to figure out, like, what can we do here? There's no data. It's a new pathogen. There's literally no data on it. So we had to come up with a new way of using the data we had available to us. And for those of you who aren't familiar with the way SARS-CoV-2 works, it basically binds to a receptor on host cells and humans as well. The receptor is called ACE2, and ACE2 is found across lots of vertebrates. Lots of things with backbones have ACE2. And so what we did was reason that the virus, well, it has to be able to bind to ACE2. And so if you have ACE2 and you have a structure for ACE2 available, a sequence for that structure available to you, then maybe we could train an algorithm to recognize when the virus will bind strongly to that structure. So that's what we did. And then we trained the model to also recognize the features of those strongly bound species. And the model made some predictions that were kind of heartbreaking, actually, because they were really rare species, or species that were already threatened and endangered. For example, the snow leopard. And then species that were really common that you really don't want to see at the top of your COVID prediction list, like white-tailed deer and red fox. And these guys, and deer mice, right? These guys are on the slide because they've all been verified since the publication of our predicted results. And the snow leopard actually died in a zoo from COVID in the US about several months after we made the prediction. And I remember presenting these results to some colleagues of mine at a scientific meeting. I think it was a DARPA meeting. And I felt kind of sheepish, like, snow leopards are at the top of our list. I don't know what that means. And then, a few months later, snow leopards are dying in zoos. And then we have confirmation that deer mice don't just get infection, but they pass it on. And of course, now we know that white-tailed deer also are passing it on to each other. And we know that American mink are also dying from COVID infection. So it's not a good set of results, but it's better to know, and it's better to know and then be prepared and figure out your action plan. So that's where we're at with COVID.
And we're not going to go out and kill all the white-tailed deer, mice, American mink, and red fox. We can't do that. I think technically it would be impossible. People have tried to eliminate some of these species and failed miserably. But I think fundamentally, when you first drafted that paper, your lab drafted that paper, I remember reading it and being more depressed about the pandemic, because it was really clear it's not going away. Not only is it circulating in human populations, but as a zoonotic disease, it is now in other species, and it's evolving in those other species, and inevitably it will eventually spill back from one of them to us. And so,
actually, it's we think about animals as like being these things that can evolve new variants, and that might be true, and that might be happening. But we also see the opposite end of the spectrum with white-tailed deer, for example. They've become these sort of stable reservoirs for variants that are no longer common in the human population. Right. So I think Alpha and Delta are now stably found in white-tailed deer, and they're just alpha and delta are hanging out in deer. And what does that mean for us, for especially those of us who aren't vaccinated?
Right.
Yeah.
Everybody get vaccinated. Why don't we, when we were talking yesterday, I realized that mid-November, where we are now, is when the first identification of SARS-CoV-2 happened in China. And we are now officially in our third year, into our fourth year, right? We are ending our third year of this pandemic. So what have we learned, since society is now laser focused on this issue in a way that it wasn't 3 years ago? What have we learned that we didn't know in early 2020 or late 2019 about predicting pandemics?
Gosh, so much. Where do we start? The fact that we didn't have our data act together was really clear. And what I mean by that is not that we didn't have data on a new pathogen, because how can you expect to have those data, right? But the mechanisms, that the organization of the data that are needed to act quickly in an emergency, those fault lines were really obvious. So we didn't have basic data on like our infrastructure, for example, let alone the data that we always knew were missing for animal species that we needed to know about for the disease, to predict where the pathogen was going to hang out in animals. So there's a lot of data gaps that were exposed during the pandemic, which, again, it's better to know where your fault lines are so that you can reinforce. I think the thing that really has changed the way that I think about my work is the importance of social behavior, right? So even if you do a perfect job of predicting everything, you can't change somebody's mind about what actions they should take, then what, like it really kind of cuts you off from making progress and preparing yourself better for the future, and being prepared. So I think human behavior, understanding how we need to communicate the results, how we need to like socialize the idea that scientists are constantly updating our knowledge about a system that is complex and evolving quickly. Those sorts of things that scientists take for granted, right? Of course, we don't understand everything about a system. Of course, we need to revisit our assumptions. Of course, we're always updating our model results. The way that we communicate that uncertainty to the public is actually very consequential, especially in times of emergency and fear. And so that was something that I took away from the pandemic and continue to ruminate over now as I continue to do work in this field. And I think health communication, it's just so undervalued. How do you talk to somebody about something that's scary, like a new vaccine?
Right.
And then how do you talk about ecology and the importance of maintaining your environment and all of those ecological interactions that folks may not have top of mind when they're confronted with a new pathogen from
Yeah, and I think there's a metaphor with climate change where climate change became very political. But if you talk to people about floods, if you talk to people about drought, if you talk to farmers about unpredictable weather, right, you could make real progress. And I was at Yale this afternoon, and somebody from the climate communications program was giving a talk, and it's really clear that there are good ways to talk to people who don't want to hear what you have to say, and you have to think different, right? I mean, steal from Steve Jobs, right? But you've got to think differently about how you communicate things, and start with the assumption that people will understand that people do want to be healthy, that people do want their kids to be healthy. And, for many, many years, standard vaccinations for rubella and other, measles, chicken pox and all these things were mostly taken for granted. And I think part of the challenge, as you said, is it's scary when you have a brand new, not just a vaccine, but a whole new type of vaccine. And how do you talk to people about, measuring risk and how we develop these things and why they're safe, right? A couple 100 million doses later, it's easier, right? But it's still not inconsequential. So I think we are learning. Scientists, we're not trained to do this, right? It's not part of our graduate program on how to talk to people about difficult issues. And I think increasingly we need to learn how to do that. So I think that's important. All right, so one of the questions, maybe the first question I ask you out of the questions from the online viewers will have to do with what you can do with to be a disease ecologist or help disease ecology. And sort of what we're asking now, or what I'm going to ask is, because we have a slide for it, is what can people do to help change this? We talked about communications and listening. I think listening well and understanding why people are hesitant, why people are scared. But what else can we do to help make it better for the next pandemic?
Yeah, I kind of had a hard time thinking about this question, but the first thing that came to mind was the fact that there's so many people on an evening webinar is kind of amazing to me. I mean, it's 7:40 on a weeknight and folks are hanging out online listening to the talk about pandemics and artificial intelligence and like nerdy stuff. I love it. I think all of us should lean into that curiosity and talk about it more with those who don't attend webinars like this, right? Like we're maybe a special group of people here, but just being more willing to engage in conversation with your neighbor, like, oh, I just heard this really cool talk the other day, or I learned this factoid about white-tailed deer, or I think that really goes a long way to socializing the idea of science and it's how central it is to societal issues.
Practice passionate curiosity.
Yeah, that's a really great way to put it. I think that's how it starts. And then, the usual, I'm going to say, like, vote for the issues. Like, this week we had on the ballot, like something like a $4.2 billion Environmental Bond Act, which is just whenever those opportunities come up, just vote for them.
Yeah, these are investments in our future.
Yeah, they are. And, when you look at the bill, you don't immediately associate like updated infrastructure for wastewater to infectious disease, but it's all connected.
Right.
Truly.
Well, and one of the best forms of surveillance during the pandemic turned out to be wastewater. Go figure. And who would have thought?
Yeah, I'm going to say that I'm going to let the audience in on an idea that Dr. Emma Rosie and I recently wrote up a paper about, and that is the fact that there are high concentrations of antivirals in our water, in our wastewater, because antivirals pass right through people and the active compounds do not get metabolized and they do not get removed from wastewater treatment. And if there's no wastewater treatment, it goes right into the fresh water. And those antivirals bioaccumulate in insects, and bats eat insects. So you can like kind of do the math in your head.
Get immunity to viruses. And
I mean, it's like, what happens when you don't take your full course of antibiotics?
Right.
Right. What happens when you don't take your full course of antivirals? So what happens when a bat gets a subtherapeutic dose of an antiviral? Literally nobody knows. So,
I was going to say, we don't know.
Literally nobody knows that. So that's like the in a nutshell of our paper, that it's a preprint, but it's out online. It's not published yet. But, like I said, it's all connected. The wastewater treatment infrastructure does matter.
Because many of these things aren't filtered out of wastewater either, because we never thought to. Right?
No, yeah.
Nobody thought that, personal care products, pharmaceuticals, right? And I guess if you do, reverse osmosis, you might get some of the smaller particles, but your standard filtration systems don't pick those up.
Yeah, they don't.
So that's why, okay, but let's tie that, let's put a bow on that. That's why it's important to lobby your state representatives, your federal representatives, to fund, upgrading the 100-plus-year-old infrastructure that is at the core of wastewater treatment in the United
really
States.
Important.
Yeah, go figure. And, I think the other thing I will always say is, be kind, right? Be supportive. When somebody is disagreeing with you, listen to them.
Yeah, I mean, I think you're right about that, because I think that we all want to live and thrive. I mean, we share that in common, right? It's just that we have deviating views on what it means to live comfortably and to thrive. But I think the pandemic has taught us that, I mean, we all don't want to get sick.
Right.
So there are common ground. There is, I mean, we all get triggered and get angry about things that we don't see eye to eye on, but at the end of the day, we have a shared things in common that we can get behind. And I feel like we need to keep that in mind in a tumultuous future.
I'm going to start with what I thought was going to be the last question, but I want to make sure we have time for it. It came at us in two different directions, one of which was, we've got a lot of students in the audience. and I'm glad for that. Yay.
That's great.
But what are the pathways to becoming a disease ecologist? We had somebody say, I have an MPH, or I have a master's in public health. What can I do with that?
Oh, gosh, so much. I mean, a master's in public health has trained you to already think in a certain way.
Right.
So kudos to you. But I would say for anybody who's interested in leaning more into disease ecology, especially with respect to zoonotic diseases, it is, I will say that it's easier to get into it now. There are actual programs that you can like get a PhD in or go get a master's in that did not exist when I was a student. But I think across the board, it requires you to bring your creativity and your perspective to the problem. For example, the machine learning and AI angle is never something that it was not a trodden path. But when you become familiar with the question that is niggling at your brain, and you say, what's a solution to this? How can I tell the difference between animals that have disease and those that don't? Those are questions that you recognize for a reason, because you're you and there's only one kind of you. And we need all of the creativity that we can bring to bear on these questions, because they are so complex and they require us to reach across disciplines and think about solving problems that are really complex from multiple different angles. And so for students who are interested, I would say, don't feel like it's an unapproachable field. It really does require your creativity and gusto and commitment.
Well, and I think what's fascinating about the work you're doing, and you're working with people at NASA, you're working with people at IBM, you're working with people all over the world. And I think one of the things that's really interesting to me is, with all of those technical skills, the point you made about what you learned from the pandemic is how social this is, right? And how important social behavior is.
Mm-hmm.
And I think one of the things that many of the scientists at Cary Institute spend a lot of time on is the social ecological interface, right? And we know that whether it's looking at the way that freshwater fisheries are managed, and the work that Chris Solomon does, or the work you mentioned that Emma does, on humans and the pharmaceuticals and personal care products we use, and how those influence primary productions in our streams and river, right? What the interaction is with microplastics. So all the work that was done in Baltimore and in West Baltimore on how people interact with their ecosystems, how our behavior affects the mosquitoes that are present in those ecosystems, and how poverty intersects with this. There are so many different fields of sociologists, social workers, anthropologists, economists, right? Modelers, computer scientists. I think the question is, what are you passionate about? And then how do you apply that to something you care about, right? So what's the technical field that moves you and interests you? And now these days we need everybody.
Yeah, absolutely agreed. It's like, if we, that I don't know which philosopher, I'm imagining this image in my head of like a statue that's really big, it's covered with a sheet, and you don't know what's under there, but if you like reach in and like you tug on it, you're like, oh, I think I feel something that is round, and or you like have a tool and you poke that shaped hole in the cloth, and you keep doing that and you see, oh, I see a picture from my angle. Well, all of us have different tools, and all of us are standing from different angles and looking at the same picture. And I think it takes us communicating with each other and drawing from each other's strengths to really get a full sense of the picture. And even if we can't get a complete sense of the picture, at least it's better than what we had from our little corner with our little tool.
Right. And so I think the answer to what can you do, I think the answer is do what you love, right? So data visualization and graphic design and art, and the way art informs science and talks to people about ideas, this is a whole new field that's maybe 20 years old.
Oh,
right?
yeah. And it's more critical every day. Yeah. The communication bit of it, I cannot emphasize enough. I wish that I had more training. I wish that I had the art skills that some of the folks that I've interacted with have, who wish that they had science skills. And I'm like, no.
There are lots of scientists. Right?
If you're a great communicator visually, oh my gosh, the world needs that.
Right. All right. So there was a question from somebody, which I'm going to try and phrase slightly differently, but they were asking about, how did you find out that the deer mouse was, potentially a COVID reservoir? Was it, was it chance? What about the white-footed mouse? But generally speaking, can you just talk about the kinds of biological parameters that you load into the model, and then how those essentially get churned through AI and spit out, or through machine learning and spit out as these species are the ones we worry about?
Yeah, it's a bit like giving the computer lots of examples that are different. So if you have a list of like 2,200 species and you have a bunch of features for each one.
How many kids they have in a litter, how big they are, how big their kids are, right? How long they live.
Yeah, their metabolic rate, what they eat, all of those kinds of things. You have, columns and columns and columns of features that together paint a picture of that species for you. So you have that kind of structure that you are then training the algorithm to identify, but what you're doing is you're labeling some of those as, yes, you carry disease. Yes, you carry disease. And that disease can be a filovirus, or it can be anything that's zoonotic, or it could be a virus that is SARS-CoV-2, right? So you can set the labels, but you're giving it a couple of labels, and you don't have all the answers to every single species in that data frame, but you have some of the answers. And what the algorithm is really good at doing is recursively learning and taking its computational power and learning from that high dimensional data set, all of those columns considered at the same time across all 2,277 rows, and telling you after hundreds and hundreds of decision trees, the combination of those, the ensemble of those says that the ones are different from the zeros, the carriers of disease are different from the non-carriers, because of these features.
Right. And then it starts looking at the other species for those features and says, okay, this group is more likely than that group to carry the disease?
It could do, yep. There are many different ways that, it's the way that you set it up, the way that you as the scientist tunes the model, but that's essentially that's one way that you could use machine learning to make those distinctions.
All right. So here's another one. So are the kinds of data you collect going to be facilitated by things like iNaturalist? I know some of the data might be, but how would, so for those of you who don't know, iNaturalist is this wonderful program on your app on your phone, which allows you to take a picture of something and then it will mostly tell you what it is, right? And then of course it's geolocated because it knows where you are, and so you've got a point location. Yeah,
it's time-stamped, it's geo-referenced, and sometimes an expert will go and look at your picture and say, yep, a couple of experts will agree that that is the species that they say it is, and then it becomes research grade. So there's a high level of confidence associated with that record. But those kinds of data points are really valuable because for species that are really widespread, it can really help to have a picture of what they're all doing at different times of year and how many of them there are in different places. And so one example that used iNaturalist recently in my group is we were trying to figure out what are the conditions that cause a food plant to flower. And the flowering is important because the fruit bats presumably are seeking these resources which become available at different times of year. And the time of year where it's most critical for the flowers to be available is during the winter. And so if these bats don't have this food available to them in the winter, then they start to forage in places where there are people, the food sources are suboptimal, and nobody's doing great. So it's part of a larger hypothesis that resource limitations are responsible for bats being in closer contact with people and also bats possibly shedding virus that could spill over into humans.
Because they may be stressed.
They might be, nutritionally stressed because they're not getting the resources that they need during a time where they're trying to thermoregulate because it's cold, and also there's not a lot of food. So there's all these stresses that they're trying to weather. And so the idea was, well, if we can try to understand what's predictive of the flowering, then we can try to pull that thread and follow the trail to the ecology of the bat. And iNaturalist was actually crucial. There weren't enough records for us to be able to extend it through all the seasons, but we trained an algorithm to recognize from these research grade images of eucalypt flowers, which ones are flowering and which ones aren't, and what times of year, because you have the timestamp. And if we had had more records, we could have maybe gotten it to where it worked great. It was like 97% accuracy or something. It's very high. So the algorithm had no problem telling the difference. What we had a problem with was the length of the time series was not quite long enough. We didn't have quite enough records over the amount of time that we needed. But it was just one example of how citizens can,
Well-
well, we used to call it citizen science, but
Right. The power of phones. I mean,
Yeah.
the computing power of phones is remarkable. And I think what you're really saying is we've gone early in the history of when we call things, citizen science, and it's now community-based science, or but early on there was a sort of a derogatory tone to it sometimes, and it's like not real science and stuff. But I think
it's crucial.
But I think what we've discovered is that this raft, this army of people out in the field with their phones taking pictures of things and uploading it, and iNaturalist and other databases being able to churn through that and give us information has really changed the way we see the world, right?
Yeah,
And so
yeah.
I think it is important. All right, so I'm going to there's a very long question, but I'm going to take a piece of it. So this is really a question for Rick. I'm seeing if Rick or Felicia's on, because we can mute them. But we're in a world in which we are fragmenting habitat, we're losing forest blocks, we're interdigitating people and wildlife, and we're reducing biodiversity. And how does that relate to disease, pandemics and disease spillover? What's the relationship between biodiversity and health? I mean, essentially.
Yeah, it's a complex relationship, and there's not a one-size-fits-all answer to that question. The system that we're probably most familiar with is Lyme disease, because the story there is that by reducing biodiversity, you're actually leaving behind the species that do quite well at amplifying the Borrelia pathogen, and also allowing for the ticks that are responsible for feeding on humans most frequently to persist in high numbers. And so that has been studied for decades. The ecological data are so solid. They're the studies that inspired me to go into the field that I'm in now. But there are other systems in which the answers to that question are quite complicated, right? Because in some cases you have higher biodiversity, and that means you might have more individuals of a species that is actually super competent for a pathogen. And then you have to think about what are the interactions between the species, and how do those interactions change when a development moves in and changes the interactions. So the spillover interface tends to be where there's contact, right? Because you can't get a zoonotic pathogen confirmed if people aren't there to get sick and tell you that there's a zoonotic pathogen. So the human contact interface is important, but it's the ecology of the animals that are there that are the reservoirs, and how their abundances and their interactions are changing that are super important. So the biodiversity question is crucial, but it's also complicated.
Okay, I'm going to do two more questions and then we'll wrap up. Somebody asked, do the algorithms feed back to adjust for data gaps or anomalies?
Yeah, that's a great question.
And how does that work?
Yeah, so it works differently for different algorithms. One example I can give is that in the algorithms that we have used frequently, they actually treat missing data as a feature. And so the algorithm is trying to split the data that you give it, into two homogeneous groups, so that the values of the two groups are more like each other and more different from. They're splitting on differences, and then the guys that are in the same groups are like more similar to each other. And if a bunch of species are missing the same kind of data, they get put into a group on their own. So what that ends up doing is treating missingness as a feature to learn on, which is really important because, as you know, rare species tend to have more traits missing for them in a lot of cases. Species that are small are harder to trap and harder to study, species that are secretive. So these species are missing trait data, but they're not randomly missing. They're missing in a way that's meaningful. So the algorithm deals with missing data in that way. And there are many different sort of versions of that. But yeah, it does deal with missing data.
Right, and that, there's a follow-up question to that, which is, do we need more ecologists and naturalists to collect these data?
Yeah.
Yeah, and I think the answer, that's an easy one.
Never going to say no to data collection.
Yeah, and these are, as you say, the common species we've got a lot of data on.
We do, except with the Zika study showed us that actually primates that are the most common and actually considered to be pests are missing some of the most important data. It's like we don't know the traits of species that are in our own backyard. It's kind of shocking.
Well, so we're going to have to start a collaboration with all the natural history organizations around the world and say, we need data on the macaques, right? Rhesus macaques, and they're the most common primate out there. But could you please?
Not a bad idea.
Yeah. All right. So here's the last question. A friend in Millbrook actually says, okay, I'd love to know, in the wake of COVID, what keeps Barbara and her fellow disease ecologists up at night?
Gosh, that's such a good question.
Then I'm going to throw you a softball after that.
It's not the ecology or the technology that feels limiting. What keeps me up is our complacency and whether that's going to rule the day.
Right.
Right. So everyone says, oh, things are going to be different now. We've had COVID. It's a learning experience. And is it going to be a learning experience?
Monkeypox wouldn't suggest we learned a lot.
I don't know. And are people gonna, so say we had a pandemic next year, like, are people gonna say, oh, we've been through this, whatever, mask, I'm not gonna eat, like, are we gonna snap to attention and we know what to do now? Like, Hong Kong was so good at this with SARS 2 after SARS 1, right? They knew that masks worked, and it wasn't a social taboo to be wearing a mask. It was just something that you did.
Yeah.
And that's what I worry about the most, I think, is not the technology. I feel so optimistic about the collaborations with computer scientists are amazing. And with virologists, I mean, I think all of the motivations are there. It's the complacency and our inability to sort of map this onto social behavior and how to do the communications right. That's what keeps me up at night.
Great. And I know you're optimistic also about the future vaccines and the ability to target vaccines at broader groups of diseases and classes of diseases. So we have learned a lot. But I think we'll end with that. And Barbara, thank you. The emphasis on the social ecological, the social science piece of this, I think is really important, and the public health piece of it. We have to be better the next time at communicating and listening and understanding. So that's great. I will just say goodnight to
Thanks to everybody who
couple hundred people who came, and the 138, 36 of you are still here.
Wow.
Thank you for coming. Make sure we're going to send you out the link for this. If you like the seminar or the webinar, please, as Barbara said, pass it on to your neighbors and your friends and your family. We don't charge for these, but we love it when we build our audience. And we've had an audience all over the world. So thank you for coming tonight. For those of you who got up early in Asia, thank you. For those of you who stayed up late, parts of Latin America and Europe, thank you. And Barbara, again, thank you for a great conversation, and have a wonderful night.
Thanks, everybody. Take care.
Bye.